Hey, what's up?
Not much. How are you?
I'm all right. Of
It's good.
course, my internet decided it needed
Mm.
to go out
Yeah.
for, but we're here finally. So
Mm-hmm.
now let me get everything booted. Good. Um, so let's see. All right. So, we went over last week. We went over metabolic
Mm-hmm.
uh pathways and stuff.
Yep.
Um, so we went all the way through to basically we went through glycolysis, gluconeogenesis, and then cellular respiration.
Mm-hmm.
So now can we go through what is it that I wanted to see here that I saw from Monkey? Um, there. So just to get a grasp, should I be going over the like where should I be starting with the psychology sociology area?
So you haven't taken those classes, right?
I didn't need to take
Yeah.
psych
Yeah.
or soc in my stuff.
Yeah. So you can learn that. Yeah, you're gonna There's a. I think it's like some type of like. Um. Oh, let me see one sec. Yeah, there's like a big Khan Academy, like 300-page plus document
Yeah,
with everything in it.
it was my best content diagnostic actually. I scored like a 55% on it, so it wasn't
Oh,
bad.
nice. Yeah.
I was just making sure.
Mm-hmm.
Okay, so I'm good. Okay. So since we're still going with bio here, for a little, I wanted to, yeah, I think this is it. Bioenergetics section. I scored poorly on. And what are the type of questions here?
Okay, so I think this is the link for it. I'm putting it in the. Oops. I'm putting it in the chat. And Anki, of course, also for learning all of the psych soc stuff.
Yeah. Okay, cool. Wonderful. All right. So, work like that. All right. Awesome. Now, okay. So, yeah, bioenergetics. That's like one of the areas that I scored poorly on, like completely. So I wanted to like go through, like here, let me share my screen so we can
Mm-hmm.
go over the question,
Yep.
the questions regarding that. How do I do this again? Okay, there you go. Boom. So yeah. Um,
Mm-hmm.
okay. So yeah, we got Is that starting from? Yeah, I believe so. Yes. Okay.
Yeah.
So yeah, here.
So, so remember when we last time when we met, we went over in the metabolic pathways the irreversible steps.
Yes.
So the irreversible steps are when you have a negative delta G,
Yes.
and
I
that's
remember
exergonic.
that. Mm-hmm.
So, so yeah, if they're talking about um, the rate limiting enzymes, they're talking about the irreversible steps where the enzymes, function. So you're not gonna have. So you can either have like a delta G that's greater than zero, which is positive, and that's endergonic. You can have
Mm-hmm.
a delta G that's less than zero or negative, and that's exergonic.
Yes.
But if you have a delta G of zero, it's just pretty much like at equilibrium, so it's not going to go, in either direction.
Yes.
So, so yeah, some clues here are rate limiting, right? Regulation, right? These are the
Yes.
things that these enzymes do. So yeah, since since they're talking about the rate limiting enzymes that are targets of regulation, we
Mm-hmm.
want to take our knowledge of the fact that it's going to have a negative. Hey, it's going to have a negative um, delta G. Um,
Okay.
so C and D should be out because it basically says that it's zero.
Mm-hmm.
And now this thing over here, this K, you're gonna see. You're gonna see my cat keeps like eating my plants, and then she like just throws up later or something. But so okay, the all the K stuff. There's gonna be a lot of them like K equilibrium, like here KSP, KA, KB, KD, uh, you know, tons of them. But they all follow something called the law of mass action, which means that it's essentially going to be like the concentration of the products over concentration of the
With the
reactants.
reactants. Okay.
So if you think about it, so let's say that we had equal amounts of products and reactants, what would this ratio turn into?
If we had what?
If we had equal concentrations of product and reactant.
It would be one.
Good. And then what if it's greater than 1?
Um, then the K would be greater.
Uh-huh. So I guess what I mean is, um, what would happen to the concentration of the products and the reactants?
Oh, if everything was to go up,
If K is a number that's larger than 1.
then the concentration goes up.
Yep, but for which?
It would go if K is more, then it means that the reactants are higher, right?
Well, reactants are the
Or oh no, the product would be more concentrated.
yeah, because that's on
Okay.
top of the
Yeah,
ratio,
because
right?
that's okay. That's the
Yeah.
deciding factor. Got
Mm-hmm.
it.
So yeah, if the K value is larger than 1, you have more products than you have reactants. And then if it's less than 1, you're going to have more reactants than products. So for this question, we want to think. Well, we could eliminate C and D, and then A and B are both correct in the fact that delta G is less than 0. But is it going to be something with a higher KEQ of like a KEQ larger than 1 or a KEQ less than 1? So, so how would you reason this out? I mean, there's going to be like, well, yeah. How would you reason this out?
Okay, so if they're saying that obviously the delta G is going to be they're saying that delta G is going to be less than 0, which would make sense. But then the K equilibrium, if they're talking about rate limiting enzymes with metabolic pathways or often targets regulation, these enzymes generally catalyze reactions that feature which characteristic. So you would want a higher concentration of products in that case, since it's regulation. If you don't have
Exactly.
enough products, then it's going to be non-homeostatic.
Yeah, perfect. Yeah, I was gonna say that there's the one way. Well, there's like a formula that I can show you, but there's also a way that you can reason it out, and you want to have that type of skill on the test. So you
Yeah.
did a very good job. So yeah, if delta G is negative, the reaction becomes spontaneous, and that means the reaction moves forward, right? And if a reaction moves forward, it's going to have more products than reactants.
Yeah.
So, so yeah, and then there's also a little equation here, which is delta G is equal to negative RT times ln natural log of K. And let's talk a little bit about about logs. I have some stuff in the shared PDF folder about like math, but what do we think of? Yeah.
This one.
Yeah. So
Yes.
So over here, I'm gonna clear this. We have these on the right. We have the log, these types of log rules, right?
Yes.
For instance, if you're so a log is basically like an inverse of an exponent. So, so the exponent laws kind of show themselves in like logarithms. So like for instance, if I had x to the power of a times x to the power of b, what would this end up being?
XAB.
What's that?
XAB.
Well, it would be x to the power of a plus b.
Plus B. Yes.
Uh-huh.
Okay.
Now, what about x to the a? It looks like a 9, but x to the a over x to the b.
Would it be XA minus B?
Good. And what about x to the power of a that's taken to the power of b?
Then that's when you say XAB, right?
Yep. Good. So now, if we look at logs like this one over here, this is log of a times b, right?
Yeah.
So it's essentially what we're doing is we're going back this way. So, or rather, so if we have log of a times b, then that just turns into like adding it. So log of a plus log of b, and then dividing it is just subtracting. And then this one is a little different because if you take the log of something that's to the power of something,
you times the power of something to the log.
Yep. So yeah,
I remember that from biostats. Yeah.
So yeah, that thing would come back down there. Yeah. So yeah, so that's that. But let's think about let's see. So okay, so let's think about. So log of, oh, I guess I wrote it here, but log of 1 is 0, right?
Mm-hmm.
Okay, now let's think about what if we have a log of a number, let's say like, n, where n is, less than 1. It's not negative though, so it's greater than 0, but let's say it's like 0.5, right?
Yeah.
What, how would we be able to take the log of 0.5?
Ooh. Okay. Well, if the log of 1 is 0, then okay, I got it. I got this. So can't you just make it into a fraction of some sort?
Yeah, that's exactly what I was gonna say. I mean, there's, you could use.
You can put it over 1, no?
So
Or
let's think about it, right? So
over
this thing
itself?
is 1/2, right? 1/2.
Yeah. Yeah.
So that's what we can do. We could take log of 1/2, right?
Yes.
And you see how this is essentially, well, okay, what do we do from here?
Um. Well, log of 1/2, which um.
Does it look like any of these things here?
Yeah. Okay. So you would just make it negative,
Oh,
or
what's the thing that you see that that you were looking at?
I was looking over here.
Oh, okay. So there's a 1. Well, that's actually a good point because there's a 1 there. But let's say we didn't have a 1, right?
Like
Let's
you're
say
trying to get
that.
at this, right?
Yeah. Cause
Okay.
yeah, cause so yeah, we can do it in two ways. We can do it like log of 1 minus log of 2, and since log of 1 is 0, that's why
Yeah, that just makes it
this
the log
thing
of
over
2.
yeah, that's why this thing over here is just the negative log of a.
Yeah.
But what you can also do is you can just kind of leave it as the decimal, or rather you can take it or put it in the scientific notation. So like you could say it's log of 5, times 10 to the negative 1, power, and
Yeah.
you could do it that way also. So okay, so we have that now. So we and then I guess I should say that since this turns out to just be 0 minus log of 2, right? This would turn into a negative number, right?
Yes. Would turn into a negative number.
Yeah. So if we
And you can
have
have a negative logarithm, right?
what's up?
You can have a negative logarithm, right?
Oh no.
Yeah, right?
But yeah, you can't have a negative log, but you can have a log of some quotient or something that when you do the log rule of subtracting it, you can give you a value that's going to be something that's negative.
Yeah.
So when this number n here is between 0 and 1, aka like a fraction, this entire value turns into like a negative type of value.
Yeah.
And then the opposite would be true if you had, let's say, log of m, where m is larger than 1. Then you'll get your positive number.
Yeah.
And if you're, if that number is just equal to 0, then you're just going to get, log of 0, which is just 1.
Yeah.
So to bring it back to that question with the delta G, that's where we have delta G is equal to negative RT ln of K. Now, since we want, in the context of that question, since we want a negative delta G, we want to keep this negative here, which means we don't want to make this natural log of K. We don't want to make that negative. Because if we made this value, if we made K less than 1, then the logarithm of K will be less than 0. And that would mean that you have a negative here times a negative here, and that would eliminate, that would cancel out the negatives. So, yeah. So yeah, that's just a way of being able to do this with formulas, but it's good that you also had like the reasoning down of, the reaction moving forward.
Mm-hmm. Okay. Got it. So let me just screenshot. All right.
Yeah, so I realized that what you asked me, I think I, didn't say like the complete thing. Like, you asked me if logs can be negative. Like, a log value, like you can't take a value of, you can't take the log of something and get a negative value, but if you have a negative in front of the log, it can be a negative value, right?
Yeah.
So, yep. Oh, and then on the sheet I have like a couple of log values that could be useful to know. But yeah.
Yes. I see him here.
Mm-hmm.
Okay, perfect. Yes. Oh, Henderson-Hasselbalch equation over here.
Mm-hmm.
Is that HH?
Yep.
Cool. Okay, so coming back.
Mm-hmm.
Oh, loudness. Yeah, I was seeing that they had what do you call it, questions based on decibels and everything like that.
Mm-hmm.
Auditory math. So.
Yep.
Okay. So let's see.
Oh, this is a new one. Okay. Okay. So.
Okay, I get this one. Has a slightly positive change in group C and G, however, the reaction still tends to proceed on their opposite. Yeah.
So
the excess would make sense.
So this is a good time to talk about. I don't know if we talked about this before, but thermodynamic, like quantities, versus kinetic quantities.
All right. So no, I don't think we've gone through it. So let's do it.
Yep. So let's say that we just have, some type of reaction diagram like that, and let's say this is the reactant, this is the product, and then from here to here, I could say is the enthalpy or the bond energy of the reactant.
Oh yes. Okay.
We did
coming back.
And then this, I shouldn't have put that. Okay, so.
Enthalpy will be here. Wouldn't this be the entropy?
So these will all be enthalpy values. So this is just this is the enthalpy of the reactant. This is the enthalpy of the product. And then the difference between the two over here is called, well, it's the delta H, which is the enthalpy of the reaction as a whole.
Yeah.
Okay. Now, things that deal with overall, so we see that the reactant is at a higher energy than the product, and things in nature, they want to go towards do they want to go towards higher energy states or lower energy states?
I want to go towards higher.
Well,
Right.
think about it. Is it harder to wake up than fall
Oh,
asleep?
it's easier to sleep than to wake up.
Yep. So things in nature want to move to or to lower energy states because they're going to be more stable that way. So the reaction that I just drew shows that the energy level of the reactant is larger than that of the product. Now, if that's the case, why do we even have reactants? Why haven't all the reactants, in the world or the universe just turned into their more stable products?
All products would have to exist all the time, right?
Yeah,
I mean,
sure,
yeah, basically.
but for any given reaction, why if it's so much more, if a product of something is so much more stable, what's the deal with the reactants? So, what in this diagram have I not marked yet?
Well, we haven't talked about the K capacity over here.
Yeah. So this here is going to be. Let me use different. Is going to be the activation energy.
Yes, the activation energy is the empty space over here. Uh-huh.
So it's the hump essentially.
Yeah.
And although it's called activation energy, we don't really think about it in terms of energy. We think about it in terms of so kinetics is all about speed or rate of a reaction. So for instance, so first of all, the reason that not all reactants have turned into products is because you need significantly more energy than the reactant already is at to move forward. Right? So so for the most part, reactions, especially the ones in our bodies, take forever. Like they would take like tens of thousands of years, but they don't take forever because we have enzymes. Okay? And enzymes act as catalysts.
Yes.
Which means that enzymes will lower that activation energy, helping this reaction move forward. Okay?
Yeah.
So, thermodynamic stuff deals with overall stability, and examples would be like enthalpy, Gibbs, entropy, all sorts of things that deal with, how stable something is. But kinetics deal with rate or speed. And the only examples would be like activation energy and temperature as well, and maybe like if you have larger surface area or something like that. But kinetics is just concerned with speed of a reaction, and thermodynamics is concerned with the stability. That's why in orgo, I don't know if you've seen any equations, but it would be like 50%, let's say like this product is made, and then like 30% of this other product is made, showing you that. Have you ever seen like problems like that?
Not quite yet, no.
Yeah. So yeah, there's like problems that you'll see, like it'll be something like whatever, like A turns into B, and what that exactly means.
Oh, wait, is this like questions that say what's the rate limiting step and whatever it is, or what's the activation?
Oh, so
What's
that's
the academic design of this equation?
that's more of like a Gen Chem thing. But like for instance, if this
Oh, yeah.
if a molecule turned into like two different things, and let's say like 40% is B and 60% is C, that can
Hmm.
tell you that can tell you that either like one of them, B or C, is going to be fast and kinetically a kinetic product, or something can be more stable, and that would be a thermodynamic product. They like to ask
Yeah.
they like to ask questions about that, and that can be kind of tricky.
Okay.
Yeah. So this question here, um, it's talking about glycolysis, and then it's talking about the isomerase, um, and then it says a slightly positive, delta G, but it still proceeds. So right now, like the word slowly here should make you kind of not want to choose this, because we know that speed is something else entirely. Especially considering we're talking about Gibbs energy here, which is a
Yeah.
thermodynamic thing. Um, then let's see. A says, oh wow, most people chose A. So A says that the reaction is coupled to ATP hydrolysis, and sometimes that happens, but that's when you break like a phosphate bond in ATP, and then you use that energy to drive something forward. But if that was the case, we should have uh, less of a positive change in Gibbs, right?
Yeah.
And now tell me why D doesn't work.
Activation of the reaction catalyzed by 12. Well, that wouldn't work because they say the reaction still tends to proceed under physiological conditions. So, um, you would still need energy for that reaction to go out. So why would there be small amounts of energy,
Well, uh,
right?
think about it in terms of what is it like really asking like you to think about.
Activation of the reaction catalyzed.
So do you see any? So we know from this question that the answer we're going to look for is something that's you know not going to be related to kinetic quantities. So B is out because that's a kinetic thing, the speed, or the it happening slowly.
Yeah.
But what about D?
Oh, okay. Well, yeah, they're talking about the um, reaction itself having low activation energy.
Well, so they're talking about you know thermodynamic things like Gibbs, right?
Uh-huh.
But remember the kinetic things we talked about was activation energy, right?
Oh, and that's talking about kinetics.
Yep. So,
And we're here talking about stability.
uh huh. Yeah. So D
Okay,
is
so
wrong for the same reason that B
D is wrong.
is wrong, right?
Got it.
Yeah.
Okay.
So it'll be C just because, Le Chatelier, essentially. If you have a
Mhm.
buildup of DHAP, it's going to want to turn into not DHAP.
Yeah.
Mm-hmm. So yeah, definitely remember these uh, types of values.
Okay, so kinetic deals with rate speed, and that's activation energy. And what's the other one there? See on
So,
there?
uh, temperature.
Yes. Okay, that's what I thought. I have that here.
Yeah, and then for thermodynamic stuff, enthalpy, Gibbs, entropy.
Got it. Okay.
I'll be back. 1 second. I'm gonna get some water. All right.
All right. Okay. So let's see. The index between oxidative phosphorylation and substrate level is the latter. All directions are. Yeah.
Yeah. So substrate level phosphorylation is, what happens with, like, throughout most reactions. Like, that's just, when a substrate gets phosphorylated by, like, ATP or something like that. Whereas oxidative phosphorylation is when it's going to be connected to the electron transport chain, because it'll eventually go all the way to oxygen.
Yeah.
So, yeah.
Okay.
Yeah, so... So for something like this, right? You can... So if something...
I get why it would be a reducing agent.
So, in redox, right, you always need to have something
an oxidation portion and a reduction portion to
it. So for this, right away we know A and B are out, because we're gonna need to have two of these.
Yeah.
Right.
And then look at that.
So if you get reduced, right, it means that you oxidize other things. If you get oxidized, it means you reduce other things. So, so it's you can never just have oxidation, and you can never just have reduction. So that's why it'll be 2 and 3.
Yeah, the following reaction was oxidized, which means you need a reducing agent.
Yep.
If NAD plus and H plus were reduced into NADH, then it would be 1 and 4.
Yep.
Substrate of hexokinase is glucose. Yeah, okay, now I understand that after we went through glycolysis last week. So, let's see. The rate limiting step in most smooth tissues. Yeah, K1. K1.
What's up?
Oh, that was it. Everything as well. Glycolysis takes place in the cytosol. We know that now. Glucagon. Increase the activity of this one. Yep. Fructose 1,6-phosphate. Phosphatase. Carboxylate. Yeah, I remember that. So, which statement is true? Oh, yeah, when we went over last week, I remember that. Yes. Okay. Yep. That's understood. That's understood. Just a quick question. I had put all the above because I had figured that since everything was a lipid, in this sense, membrane fluidity would be low.
Uh huh.
But um, what do you call it? Is this like is that was most likely now that I see a trick question
Mm-hmm.
and stuff because the question was really asking which one of these would be associated with increased membrane fluidity, not saying would there be membrane fluidity.
Yeah, yeah. So it's kind of tricky how they worded it.
Yeah,
Because yeah, if you read it as what's just associated with the membrane, then it could be
yeah, all the above would be yeah,
Mm-hmm.
yeah, okay. Um, yeah, okay, I knew that. Um, you see, I had thought that the cell to cell communication was tight junctions here and stuff instead of gap
So
because I figured that they went closer together because I know that the from developmental
mhm.
biology, you had to like know all that stuff about cell cell communication because that's how stem cells work
Mm-hmm.
and everything completely. But um like I know about the paracrine, juxtacrine,
Uh-huh.
and autocrine signaling and all that stuff. But I was figuring tight junctions were going to be better for cell-to-cell communication since it would just allow easy transport and access between materials between the two cells.
So you know how like cans have, like, let's say this is like a 6-pack of something, and
Mhm.
there's like this kind
Yeah,
of
the
like
little divisions.
plastic thing like that connects
Ye-
them, and so like if you want to
Yeah,
take it out, you have to like pull it underneath.
yeah.
So that is how tight junctions are. The number one thing to know about tight junctions is that uh, they're impermeable to water. So
Yeah.
they're and that's because they're tightly like connected to each other. But
Yeah, they're essentially conjoined.
but
Uh-huh.
um, you know so if they're super close to each other, touching, sure, why not? Why wouldn't they have an easy time, communicating cell to cell? Well, there could be something that's better. Like if I had a cell here, like that, another cell here, like that, and another cell here. Stuff that happens here can
Oh,
go all the way through there.
yeah.
Mm-hmm.
And then I remember a diagram from developmental bio. Would that something sorts not coherent? Uh,
Connexins or something.
yeah, connexins, right?
Uh-huh.
Is that's what it is? That allow the communication to be better then?
Let me make sure. Yep. Yeah, connexins. They're in the uh, gap junctions.
Yeah. Okay. I was figuring that the connexins would make it easier.
Yeah.
Okay. Now I understand.
Mm-hmm.
Okay. Um, lateral diffusion occurs relatively quickly and even yeah. Well, yeah, that's just right there. That's like juxtacrine signaling, essentially.
Uh, yeah. This is like kind of just how the membrane's always uh, changing, I guess.
Yeah.
Um,
Yeah. Vitals. Mitochondria, yeah. I'll take place with them. Oh yeah, that was stupid by me. I don't know why I included the smooth in the ER in that. I included the smooth ER, I think, from virology. I was thinking because a lot of viruses use the smooth endoplasmic reticulum to synthesize their own proteins
oh,
and
interesting.
everything. Yeah, it's like they use it as a transport and everything, so it's all through like viral proteins and and whatever. But that's what I ended up thinking instead of that.
Mm-hmm.
Um,
Oh yeah, you're super close here.
yeah. Rough ER has ribosomes attached to its membranes and synthesized what is called
So, what does a
ER.
ribosome do?
Uh, well, ribosomes themselves synthesize proteins, right?
Uh, yeah, I don't know if I'd say by themselves, but
No, well,
they,
yeah,
they read the mRNA and then they translate
yeah,
the proteins.
and they
So,
translated it to yeah.
hmm yeah,
Um, okay, yeah, and these smoothie are synthesized lipids.
hmm
Ah, probably going too quick. Okay. Yep, that's easy. Microfilaments are the smallest intermediates, then microtubules.
yep.
Centrosome, okay, mitosis, yep. Yeah, just their function. That's funny. That was actually a trick question.
This one?
Yeah. And I believe, what was it? Um, a microbiome test
Mm.
a long time ago, and everyone literally picked uh that flagella and cilium differentiate in their movement. Like that's what he ended up putting. He messed everyone up on that one.
Uh huh.
I was able to catch it, that it was the function of it. But yeah. Yeah, you see, so cell bio I'm not bad with at all over here.
Mm-hmm.
Tissue types, yeah. Since pseudostratified. Just a quick question. So pseudostratified, they go like, isn't it supposed to be like almost like this or whatever? Like they're like that or something? Like that?
I think something like that.
Yeah, okay.
Where this is the basement.
Yeah, there's the basement membrane and whatever.
Mm-hmm.
Okay. So then, oh yeah, okay. I picked. You see, everyone else got messed up here
Mm-hmm.
with the statistics. Yeah, multiple layers of columnar-shaped cells. Yeah, there's no layers.
Yeah, that's
Just
true.
columnar.
Actually, yeah, they go like this.
Yeah, and that's exactly what I just finished drawing, and I put
Yeah.
the wrong answer anyway.
Yeah.
That's okay.
I don't know who would choose. Like, why 8% chose that.
Oh boy. Okay.
Hmm.
Like in the bones. Yeah, Highland. Yeah, I remember that. Yeah. Okay. This is something I wanted to talk about. Did you see? So part of the chromosome which contains strata. I was thinking that chromatids did. I don't know why.
Well, what is a chromatid?
Well, the chromatid, right? You have your right, it's your chromosome or whatever.
Okay.
Right.
Now, when it turns into that, I mean, what does it do when it turns into that? So what does the DNA look like when it's not like that?
When DNA isn't in this form.
Yeah.
Well, isn't it in just double helix, like extended? Yeah.
So what?
Oh, euchromatin is supposed to be tightly packed, right?
Well, so okay, let's say that. So this is how the DNA would look like otherwise, right? Right?
Yeah.
Okay. Now, let's say that you're a transcription factor, right? You work by attaching yourself to DNA, and then replicating it. So
Yeah, like beta or something. Okay.
yeah. So what form of DNA here the chromosome on the left or the loose one on the right, which one is easier for a transcription factor to bind to?
That one.
Yeah, because it's a lot more exposed, right?
Yeah, it's loosely.
Uh-huh.
And euchromatin is usually loosely packed and hetero is tightly, right?
Yeah, yeah.
Okay,
So
it's the inverse. That's where I messed up. And
Yeah.
then I was thinking chromatid, perhaps because I was thinking of it in like this form in genetics.
yeah, so like
When they talk about this, and I was thinking like, you have like your little guy go over here.
Mhm.
But all right, that's where I messed up.
Yeah, so there's
Okay.
there's different ways of thinking about it, which can always help you on the test. Like if you don't remember, like if you think about okay, a chromosome, what does that do? The chromosome is just something that happens when a cell divides, but
Yeah.
what actually, what about like what else does DNA do? Like what does DNA do when it's not a chromosome? Well, it gets transcribed, into RNA, which then gets translated into proteins. So
Yeah.
here, they're telling us. So like, I could see how this part can throw people off,
Yeah.
but and that's
I like how 10% of people chose plasmid.
yeah. I don't necessarily like that it's still talking about part of a chromosome, but yeah, this part over here is what's really important, the transcriptionally active DNA.
Yeah.
And yeah, the euchromatin is loose, the heterochromatin is tightly packed, and when a chromosome or when it's super packed and condensed, it looks like the chromosomes.
Yeah.
This is
Okay.
they can also ask you stuff about histones here, like if you take euchromatin and you methylate it,
Mm-hmm.
you bring the beads closer together.
Yeah.
But if you then take the heterochromatin and you acetylate it,
Yeah.
it does the reverse.
Yes.
So yeah, so they'll always like to ask
DNA
questions like
methylation
that.
versus acetylation. It was funny how it took like it took funny enough like a really long time for people to understand the methylation acetylation stuff.
Oh yeah,
And
yeah.
I was like, oh boy.
Mm-hmm.
And I won't lie, it took me a little bit too. But
Which one?
once I understood that concept, everything got much easier with genetics. But uh, okay, I get it now. Cool. All right, so what was not a model of hypothesized for the method of OMS install? Oh yeah, that- that's semibiotic. Um, aging, shortening of the telomeres. Yeah. Uh, yeah, that wouldn't make sense at all.
0%.
Nobody, I love how nobody said that. Yeah, all this wouldn't make sense because then that means something's wrong with you. Bad. Um,
Oh yeah, here's a histone problem, I guess.
yeah. Why would I say removing the primer?
Yeah, maybe you thought it
Oh,
was like
because
an RNA or polymerase or something.
I was thinking what I was thinking was
Hmm.
in laboratory DNA science, the class I took and stuff, we worked with like putting primers and whatever, and I kept thinking of us synthesizing, like making our own primers
Mhm.
and stuff. And perhaps I was like thinking under the impression for whatever reason that like the DNA would remove the primer. It was just using it as an attaching point to
Yeah,
make the
yeah,
rest.
yeah.
So
I think that's natural to think because it's like the RNA primer that doesn't stay for the whole. It doesn't stay the whole time.
yeah, exactly. So that's why I was thinking. I was thinking RNA primer rather. Okay,
Mm-hmm.
that's fine. I get it now. Um, only changes. Well, yeah, AUG wouldn't change, but AAG. Oh, Okay, I see where I'm wrong. They just wanted me to translate it. Right? Or oh, it changed the AAU. It changed it to a G, but what do you call it? Okay, no, now I'm confused. Um, only changes to the mRNA transcript.
So yeah, let's look at it. So I'm gonna write them out. This. So then we got this. Oops.
Oh,
Yeah.
I get it. Oh, I feel like such an idiot. Okay.
Why? What?
My DNA science professor used to kill us on our tests about this. Everyone would get them wrong, and she just would refuse to never to not put them on the test, these ones
Yeah.
and stuff. And I just saw the thing over here about wobble codons.
Yep.
So I remember her saying wobble codons can be changed and not necessarily change the protein that's being made.
Yeah. So for this one, it seems like Oh yeah. Let me do this last one. This one says UG.
Okay, when they're right.
So yeah, the wobble. So in a codon, the third, whatever nucleotide.
Yeah, the third nucleotide is always a wobble codon.
Yeah, and that gives like you room for error.
Yeah,
there.
for improvements or changes and whatever it is.
Yep. So
Yeah.
it's like a silent mutation. So that's why, yeah, it'll be C.
Yeah. Okay, I understand it now.
Mm-hmm.
RNA and DNA. Yeah, none of the above of those. Had the additional hydroxyl
Yeah, that would be
when it's deoxy.
yeah.
Okay. In which of the following ways is gene expression regulated?
Yep. So yeah, this is one of their favorite topics.
This one was the tricky one.
Yeah, this is one of their favorite topics, gene expression, and it has to do with what we talked about with histones and stuff.
Uh-huh.
So histone acetylation would cause the DNA to become
Loosely
yep
bound, so any transcription factors like TGF beta, those types of things can all come in all over and take FGF, all those things.
yeah. So okay, so that could work. And let's look at the rest. DNA splicing.
Mm.
perhaps alternative splicing would have worked better if
Yeah.
that was the case.
But that would be splicing of the RNA, right?
Yeah, and that would be RNA splicing, so still that doesn't.
Because DNA, we're not changing anything, in the DNA. Like, different types of like gene expression is all about
Just expressing what's already there. You're not looking to change anything. You're just looking to express the
yeah. So
phenotype or
so you may
genotype,
have
whatever's going on.
yeah. So you may have a gene for something, but it doesn't. It's not the end all be all because it's only gonna make a difference if it's expressed. So yeah. So we know that DNA can, when it turns into RNA, the RNA can then undergo splicing to get the introns out. So B is wrong because it says DNA splicing instead. Why is C wrong?
C is wrong because okay, yeah, and now I feel stupid. You're methylating, which means you're tightly packing something, and it's mRNA at the end of the day.
So which one is more important as to why it's wrong?
The fact that you're methylating.
Well, if they're asking how gene expression is regulated, and we know that histone acetylation is a way that it's regulated, wouldn't it also be regulated by histone methylation?
Methylation. Oh, but you're talking about mRNA here, so that's the key word here.
Yeah. So this is wrong.
It would be DNA methylation.
Yeah. So this is wrong because of the the mRNA.
Yeah.
Yeah.
Okay.
And also, like, a lot of times it can feel safer to choose all of the above.
The above. Yeah, when you're not sure.
But you wanna have more of how should you should have your mindset geared towards looking for things that are wrong.
A specific one answer. Yeah.
Yeah, or look for look at at each question or whatever as like try to find flaws in it. Like when you see DNA splicing, you can think two things. You can think, well, I know that RNA splicing is a thing, but maybe DNA splicing is a thing. That type of way of thinking. Like I totally understand it, but you want to be a bit careful with that.
Yeah, indeed.
Mm-hmm.
Okay. But yeah, when we're talking about eukaryote, not simultaneously.
Yep. And that's because eukaryotes have organelles which can compartmentalize everything.
Yep.
But for bacteria, some prokaryote without any membrane-bound organelles,
those things would have to happen simultaneously.
Yeah, they could happen simultaneously. So there isn't going to be compartmentalization.
Yeah, initiation, elongation, and termination. Yep.
Mm-hmm.
I remember that. pRNA, that looks like this, right?
Yeah, like that clover-shaped thing.
Yeah. Something like that.
Mm-hmm.
Basically.
Yeah, this activation of the F-meth thing could be a little confusing. Like I could totally see.
Yeah, but the thing.
Yep.
Yeah. But F-met, everything has to start with methionine.
Yeah, in bacteria, right?
Yeah.
I guess for us it's just methionine, but yeah, I could see how people would choose initiation first. But AUG, it's a start codon, but it also codes for an amino acid. So, unlike let's say a stop codon will stop the translation, but it's not going to code for any amino acid.
Yeah.
Mm-hmm.
How many amino acids will the following? What was I looking at here? Yeah, 1, 2, 3, 4, 5. 27. Well, UUA is a stop codon, right?
UUA?
No. Sorry. Never mind. Okay. No, I understand where I'm at. Because if you're asking for what's coding for each over here, so you got 1, 2, 3, 4, 5. And then 6, but you stop here.
Yeah.
Yeah.
So that could be a
it.
little thing that you would have to know.
Yeah, no, I know that UGA is a stop codon. I just didn't read that. I should have done that from instead.
UAG, UAA, UGA.
Yes.
So these are pretty much the same due to the wobble.
Yeah.
And then this one, I guess, is still decently similar.
Yeah.
But yeah, this was a little on the tricky side.
Prokaryotes, but not eukaryotes, yeah, they're transition and transformation occur simultaneously. Prokaryotes rarely use splicing to modify. Yep, that's. Use operons. Yep. Lack operon, TRP operon. Okay, I had no idea what the Gigamonad model, so I just took a big guess.
Yeah. Yeah.
But now that I know that it's for operons, then that's just an easy thing to remember. Jacob, yeah. Yeah, heterozygosity. That's also with fraternal twins, right? They exhibit heterozygosity.
Not sure.
I saw that on Anki.
Oh, okay.
They were talking about homozygosity for identical twins and then heterozygosity for fraternal.
Got it.
When they're eggs fertilized at the same time.
Oh yeah. I wasn't even reading it right. I just saw the zygote part. The monozygotic would be, I think, the homozygosity, and then the other type, dizygotic or whatever.
Yeah, dizygotic. Yeah.
Yep.
Yeah. Okay. That's. That was first week of genetics. They made us memorize that ratio.
Yeah.
What is it? So yeah, you see, I went too fast with that, and then I read something later on, when I was going over my developmental biology stuff. Then I remembered that colorblindness was a sex-linked trait.
Yeah, it mostly affects males.
Carried by men, right?
Yeah, it mostly affects males because, we could say that, that it's recessive because a female could have the gene for it and could have one of their alleles. It can be a carrier, but let's say this is the father here. So let's say that, I don't know why I use these letters, but the lowercase h is the color blindness gene. So you can have, big H, X, and the regular X from the dad. Same thing here, but here, Whoops. Like these guys, the two sons, they only need one of the alleles for color blindness because they can't have another X chromosome that isn't something that has causes color blindness, but yeah, so sex-linked genes, this is like the most common example that they give you.
Yeah. Yeah, mitosis and meiosis too. Yeah, the accurate example is what they're asking for here.
Yeah, so penetrance is it's different from these other stuff, right? It's like a measure of how much of people in like a population expresses something, right?
Yeah. Okay. I got it.
Mm-hmm.
Oh yeah.
Mm-hmm.
That was easy. Actually, that's why they always tell women over 35 to be super careful with their pregnancies because the chances for a trisomy birth for an extra chromosome skyrocket by like 30%.
Yeah.
Yeah. Yeah, codominance.
Mm-hmm.
Yeah, okay. So I wanted like clarification on this one.
Okay.
1, 2, and 3. You see, it was 1 and 3. I added substitution here. Why wouldn't a frame shift mutation have a substitution?
So
Either.
a frame shift. So like each codon is 3 nucleotides long, right?
Mm-hmm.
So let's say I have A U G A G U A T C or something like that.
Yeah.
Now let me do this. So let's say I'm gonna take another A and I'm gonna put it in here in this spot.
Mm-hmm.
Then this turns into A U A, right? And then this stuff over here, I think this was a G A G that I wrote. So it would be G A G. So so this is the first codon. I'm just gonna mark it 1, second, third, right? But if I input an A after this U here, I get A U A G A G, right? And then U A T, and then just a C.
C and then dot dot dot.
Yeah, so that you just have one C here, and it messes everything up because this AUG is gone, right? I mean, like I guess theoretically this would make it not even start.
Yeah. Oh, okay.
But it's not because of that. That's just a coincidence in that I used AUG. Like it turns into something like let's pretend that this is not AUG. Let's pretend it's something else. It's gonna mess it up because it will turn into AUA, and then the next one is supposed to be AGU, but it turns into GAG. Right? It messes everything up because it
by substituting. Okay. Yeah.
So if you insert an A or anything, right, that's not a multiple of 3, it's gonna mess up the reading frame. Same thing with if I got rid of, let's say I got rid of this t, right?
Mm-hmm.
That'll mess up the reading frame as well. So the only thing that insertion or deletion will cause a frame shift mutation unless you're inserting or deleting 3 or 6 or 9 or a multiple of 3. But if you just did a substitution, right? Like let's say I take this u and I take it out, and I'm going to just take a t and I'm going to put it where the u is. It's still going to have the same reading frame, AUG. I mean, this one will change. It'll be AGT, but the one after it will stay the same as well. ATC doesn't, mess with the reading frame.
Yeah. Okay. Got it.
Yeah.
Yeah, translocation. Fine. Yeah, I see where I'm off now.
Oh yeah. So this is when you have when a mutation causes a different amino acid to appear.
Yeah.
So that's what that one is. The frame shift mutation we talked about. What can you tell me about silent mutation?
Well, we were basically talking about them earlier,
Okay.
right? So, silent mutation would basically have to do with the wobble codon and stuff, but it won't necessarily mess with the entire.
Yeah, exactly. It won't mess
At all.
it won't mess anything up. Like, even though like, so we know frame shift is a worse. Missense could be bad, but you're just changing like maybe like one amino acid. Silent mutations.
Mutations, they happen all the time.
Yeah, and they're the most harmless. But you may also have a premature stop codon, and that's what would be a nonsense mutation.
Yeah. Got it.
Yeah.
Oh yeah, I remember that.
Mm-hmm.
That was a for sure question. Okay. Yeah, I need help with this.
Okay.
I do not understand it.
Yeah. So the Hardy-Weinberg thing. So let's say that this is just the I guess like the occurrence of, let's say, P is the dominant trait and Q is a recessive trait. So this is just the allele frequencies like in any given population. But now let's think about what happens if we go back to like Punnett squares, we can kind of see how this works here. So like, let's say I have like capital A, capital A, little a, right? This thing would turn into big A, little a, little a, big A, little a, right? Okay, so now this P is going to be the dominant, so let's say that that's essentially the A here, the capital A. This Q, which is the recessive, let's say it's lowercase a, so I can draw this over to have it be something like that. Oops. Like a PQ here and then a PQ here, and when you have two of the P's, now since the P's and the Q's are representing like a numeric, like a number essentially, when we have two P's, it's instead of it just being P and P, like the A and A here, it'll be P times P, or P squared. Okay?
Yeah.
And then, Yep.
All right, continue.
So yeah, this is going to be p squared. And then let's think about the two q's here. It'll be down here, q squared. And the sizes are going to be significantly different because, unlike a Punnett square where each, I guess genotype is given the same size, square. This one functions with, if the higher p is, the lower q is, and vice versa. So I didn't make them equal here. And then these ones on the sides, they're kind of like these like rectangular bars. So this is just PQ here, and this is PQ here. Now, how do we... Okay, so that sounds, familiar with what we know about the other way to write this kind of relationship out. And that just comes from the fact that what if you had two of these? So, in other words, if you just take P plus Q is equal to 1, and you square it, right? If you square it, you would get... So, it'll be P squared plus 2 PQ plus Q squared is equal to, and then the 1 squared is just 1. And now we have, the Henderson-Hasselbalch, what am I talking about, Henderson-Hasselbalch? The Hardy-Weinberg, equation, you can now maybe see how it's related to the P plus Q is equal to 1. This thing is just that same thing squared. Instead of them being, completely random, formulas. And now this stuff, the P plus Q equals 1, like that tells you like the allele frequencies in a population, right? But this here tells you the genotype. For instance, P squared is would be like capital A, right? Q squared would be little a, and then 2PQ would be a big A, little a.
Got it.
So that's how the genotypic kind of ratio is, and then the P plus Q1 is just the allele frequencies in the population. All right. So for instance, and then for these questions, it's just a matter of knowing which, essentially formula to use. In addition to, I'm trying to look for. Oh, yeah, here. Oops. Okay, so let's look at this question here. So let's go. Go up to the question. So here, they're talking about the frequency. Oh, they even tell you that it's P, it's little p. But yeah, that's the frequency of the dominant allele. It's 60%, right?
Mm-hmm.
Now, let's think about how we can do this, right? If we now know that, so we know that P is 60%, or 0.6, right?
Yeah.
If P is 0.6, could we find out what Q would be?
Well, yeah, it's just whatever is left from 100, so 40.
Yep, so 0.4, right?
Uh-huh.
Okay, now, what are they asking us for?
A homozygous recessive. So that would be little p.
Yeah, so now they're asking about the genotype, right? And that means we're going to use this one over here.
Yeah.
Right? So, okay, they're asking... So we actually could just... We don't even need to do this entire thing. We know that Q squared would be our answer, right?
Yeah.
So that would just be 0.4 squared, or, let's see what I can do. I'm going to turn it into 2 over 5, because 2 over 5 is 0.4. I'm going to square that. So 2 squared is 4, 5 squared is 25. So I get 4 over 25. Let's see what else I could do. 4 goes into 4 once. 4 goes into 25 6 times. 1 over 6, which is 0.17. So I kind of went pretty fast there with the math, but yeah, in the math PDFs, I have certain fractions that you can like have memorized, because like when I was studying for this, I memorized I knew that 1 over 6 is 0.17. I knew I basically knew like what 1 over 1, 1 over 2, 1 over 3, 1 over 4, 1 over 5, 1 over 6, 1 over 7, 1 over 8, 1 over 9, 1 over 10, 1 over 11, 1 over 12. Yeah, I just memorized those because it's so much easier to work with fractions, because 0.4 squared could be a little annoying. But I guess it won't be too bad if you just use scientific notation. So that would be 4 to the power of -1 squared, which is going to be 16, I think times, 4 times 10 to the -1, oops, squared, 16, I guess just 16 times 10 to the -2, because if you're squaring it, you're squaring the 4 and you're multiplying the exponent here, so this will be 16 times 10 to the -2, which is 0.16. But yeah, so basically here, key thing is to be able to tell from the wording that they kind of helped us out with saying it's P here, but you want to be able to know that it would be P without them telling you, by just them saying frequency of a dominant allele. And then when they ask for the genotype, we're going to use, this one over here.
Got it.
Mm-hmm.
Okay. Genetic drift in a population refers to... Yeah. Okay, I understand where I'm wrong, but I was remembering from my entomology course, you were talking about genetic drift within insects.
Yeah, that's cool. You took an entomology class.
Yeah. And this is technically correct,
Mm-hmm.
but it's only correct in terms of physical aspects. It's not true for pretty much any sort of event, so the chance events is what makes it more sense.
Uh-huh.
Yeah.
Yeah. Yeah.
Yeah, no. Immigration or immigration is not required.
Yeah, and knowing the Hardy-Weinberg, assumptions also help.
Yes.
Oh, it's like that. Well, the choice that you chose for our previous one. Maybe you.
Yeah, I was thinking the bottleneck effect of a high population taking over too much.
I think
Reaching a carrying capacity.
I think maybe the least here may have threw it off.
And a bottleneck effect is true that it would be less likely to happen, but it's not completely true.
Mm-hmm.
But increased fitness would be the most unlikely.
Yep.
Does that make sense? Okay. So component lacking prokaryotes. Oh, yeah, that was a dumb mistake. I don't know why I thought too much.
Yeah, I can't believe only 88% got this.
Binary fission, yep.
Mm-hmm.
Okay. Circular genetic structure, yeah. That's all we used in DNA science.
Spiked.
That's viruses. And that's all these other are bacterial.
Mm-hmm.
Apologies. Not true viruses, single size 2 eukaryotic cells. Yeah, that's not true.
I'll be right back. I'm gonna use the bathroom real quick. All right.
Yeah. I didn't understand that. This one, like, I get that I'm wrong, apparently, but I was thinking that reverse transcriptase, that RNA viruses don't necessarily need reverse transcriptase to do that, because the only one that I recall necessarily using reverse transcriptase to the most positive effect for itself is HIV,
Well, let's think
being
about
that
it,
it's a
right?
reverse transcription virus. Huh?
Let's think about it, right? So, what is it called when DNA turns into RNA?
Oh, Jesus Christ. My mind's blocked, just say it.
Transcription.
Yeah, transcription, okay.
So,
Yeah.
this would be reverse transcription, right?
Oh.
And they're saying that the viruses already have RNA, but what must they need to make DNA?
Yeah, in order to infect the individual.
Yeah,
Okay.
or just help it kind of continue to survive.
Yeah.
But yeah, that's
And
the,
DNA polymerase would make sense, but it would make sense for only some RNA viruses.
I guess DNA polymerase would make sense if you, because that usually happens when you're replicating DNA, which means you already have the DNA, you're just making more. But RNA viruses like HIV don't have any DNA of their own.
Yeah, no.
Mm-hmm.
Okay.
Like, yeah, the DNA polymerase, you would need DNA. Like, that would be DNA into more DNA.
Yeah, not an RNA to DNA conversion into that.
Yep.
Yeah, I get it.
Oh yeah, this is like, like mad cow disease.
Yes.
That's what. These are super interesting, like, infectious things, because we used to think viruses were the strangest things. Because we're like, okay, is it alive or not alive? Until we saw prions, which are proteins, like, not even like they don't even have like a structure that virus could at least have that would make it look a little like any type of other animal, but they're just really messed up proteins.
Yeah, and then they cause very bad things.
Yeah. Like the mad cow disease, there's also a disease that's associated with people who consume like brains, because if someone's infected with prions, it's in their brain. So cannibalist societies have high rates of something called kuru, which is a prion infection. And then mad cow disease is because they would feed cows other cows. So
yeah.
But yeah. And then viroids are, I think, plant viruses.
Yes, those are.
And then phages are viruses that attack bacteria.
Yes.
Those are the, really like,
Yeah, the hexagonal, the icosahedral
Yeah.
heads. Yeah.
Yep.
Oh yeah,
Oh,
that
wait,
was a stupid.
what happened there?
Yeah, that was stupid of me. Some of these questions I probably was getting to the point where I was doing it in one sitting, and I like just started
Yeah, that would make sense.
being dumb because it was 91 questions at this point.
Yeah.
Rewrite the spindle fibers. Yeah, I knew that.
Oh yeah. Oh, actually, you had the right idea here. That's like the special thing about the pentose phosphate pathway, that it uses NADP and NADPH. But I think so. You had the right idea, but here you don't even need to know really any details about the pentose phosphate pathway because they're telling you that it's during the oxidative phase.
Yeah.
So if the things are getting oxidized, the other thing gets reduced, right?
Yeah, which would make sense for it to become NADPH.
Yep.
Got it. Yeah, that's fine. It's G1S, G2M. Takes place in the. Yeah, this would be a different answer now, obviously, since I know all this stuff.
Yeah.
Since now I know more. No.
Yeah. Basically, the mitochondria is where oxygen is. So everything is kind of governed by oxygen in the mitochondria.
Yeah. Then we have cyclin-dependent kinases basically run the cell cycle. Track that which molecule.
Oh yeah.
Yeah.
This is where
answer.
Yeah.
Yeah. That makes sense. Yep, progesterone. Yep, and that's exactly what pregnancy tests for in your urine.
Yep.
Yeah, for some reason, I know FSH and LH are both in the period menstruation cycle and stuff like that.
The LH spike is the thing that has result that happens with ovulation.
Yeah. You see, I had the right idea. I just didn't have the right execution. Yeah, I didn't take into account that luteinizing hormone spike during ovulation, which increases pregnancy. Glycogenolysis. Yeah, I didn't know what that really was, so I just went and took a guess.
Yep. So, what's lysis mean?
Well, lysis means to break apart.
Yep. So, breaking apart glycogen, right?
Oh, and you would do that in muscle and liver cells.
Yeah. Like muscle cells because, you store a certain amount, and if you're engaging in activities, you're going to be using it. But liver cells because that's where, all of the, like pyruvate that goes back to the liver to be stored as glycogen and stuff. So that's why it's also going to be the liver cells.
Got it.
So I think we're gonna have to wrap up soon.
Yeah. What time is it? Oh yeah. Okay. I guess we'll just go over like 2 more and then that's it.
Yep.
Parvosperm, which contains the enzymes necessary. Yeah, I had the right idea, wrong execution. I should remember that from developmental bio. But blast assist, yep. Yeah, I was very stupid here. I remembered mesoderm, but I wasn't sure if it was for digestive cells.
Yeah. I think I have, or in one of my notebooks, I have a really detailed list of these. I'm gonna see if I can, upload them.
Yeah.
No,
I do as well.
They can ask you like really, they could ask you like a lot of stuff about this that might not come, or might not be as commonly, thought of as being asked. Like I've seen questions like that. But yeah.
Yeah, I took a random guess at that. I didn't know.
Yeah, that's pretty tricky here because we do like in the pyruvate dehydrogenase complex, we do have like a CO2 like leaving, but I guess technically it's not, the enzyme isn't part of that, of the whole complex.
Yeah. Okay, I guess so. Yeah, that makes sense. Yep. Oh God. Reading that question again, I was like, no wonder I picked that so fast.
Well, what do you think about apoptosis? What about apoptosis?
Well, that's regulated cell death. But during embryogenesis, it does happen.
Yeah, like your fingers, right?
Yeah, your fingers and everything are part of it.
Yeah.
I remember in our developmental biology class, we used a dye, alizarin blue, and that basically stains for apoptotic cells in mice that are being gestated.
Yeah.
Take them out. So. Yep. Yeah, look at that. I got that wrong. And 50% of people got that wrong. Yeah, I wouldn't have known that.
Yeah, when you take a look at the Krebs cycle, you'll see that the FADH step donates its protons. Donates its electrons directly to I think complex 2 or something like that. One of them.
Yeah.
But yeah, it's the I guess it's a succinate thing. It's the succinate one.
Yes. All right. Yeah, no, I wouldn't have known that at the time. Acetyl coenzyme A carboxylase emitted by glucagon. Okay. All right, so we'll stop there for now.
Okay.
Yeah.
Let's do that. Awesome. So yeah, I would say to continue reviewing these and
yes.
And then like have like the really high priority questions for us to go over when we meet.
Yes.
And yeah. So do you want to do like same time?
Let us do not the same time next week because